Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you, SynBYSS people, for joining us the 93rd seminar, and I call it a University of Delaware Day. I came back home after giving 5 talks in California last week and visiting and meeting many great colleagues at ACS, NASA, Stanford, JBA, and UC Berkeley. And it was hectic, but great summer to meet thousands of people all over the world last 2 or 3 months. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Mark Blenner. He is a professor at University of Delaware and used to be at Clemson University, where today's rising star speaker is working. I have known him for more than a decade and we started our faculty position at the same year with similar research topics. So he is very special to me, and we have been discussing many global problems and synthetic biology solutions as colleagues, as well as EBRC members. In addition, I recently started my startup company, Moonshot Bio, in Delaware. So we are technically or officially neighbors, although we live in different states. So Mark, thanks so much for your leadership in the scientific community and educational efforts, also sharing the similar vision with me, and the virtual podium is all yours now. Thank you again.
All right, thanks Taesuk, and thanks Dave. I'm really happy to be here with both of you today. So I prepared a few slides, and let me see where we are in sharing. How is that looking? Do I need to switch screens, or is that okay?
We seeing your the other slide. Yes, it switch. Yes, wonderful.
Better. Perfect. All right. So Taesuk, I think, as he always does, asks the pioneers to prepare some inspirational words, perhaps. And so I gave this a title for no good reason, really. I called this little brief introduction "Making Friends and Building Communities." Before I get into that, I do want to plug a couple of our community at University of Delaware. We have a very vibrant and active synthetic biology community here. I think many of my colleagues have already spoken in this series already. You can find out more about what my lab does by visiting either my personal or my lab Twitter, or the sort of new UD SynBYSS Center Twitter. You can also go to my website, which is a little bit out of date, but has some good information on there. We're always looking for excited young people to join our lab as either PhD students, postdocs, or even REUs. And so feel free to contact me. You can find me at any of those places. You can easily Google my name and find my email address as well. So to start, I think it's really appropriate to acknowledge all of the many trainees that I've had the honor and privilege to help shape their careers, and sort of in the spirit of what this entire series is about supporting our youth. I think this is to me the most important product of my lab, is the people that are trained here and who use our environment to grow and become the kinds of scientists and engineers they want to be and to make the change they need to make in the world. And so just acknowledging all of the names of the students who have graduate students and postdocs that have worked in my labs, both at Clemson and at UD. I did not create a list of the over 140 undergraduates that have worked with us, but it's a really enjoyable part of this job. And the most important thing we do is train the next generation. And so I guess on that note, I want to explicitly thank you, Taesuk, for putting together this series, and for your kind of continued efforts to focus on the youth. So I want to say a few things about the kind of research that we do, just to give everyone a flavor of it. I'm not going to get into data. We want to reserve as much time as possible for Dave to tell us about the exciting things he's doing. And so I wanted to give maybe a little bit of overview of what we do in the context of the journey, I guess, of my career. And so when I started my lab out at Clemson in 2012, we were kind of coming off of the biofuels era, and I was interested in trying to make hydrocarbon fuels, and at the time, and we sort of really quickly pivoted to making, I would say, value-added products, so still somewhat low-value type things, that were hydrocarbon based. And so we looked around for how what sorts of tools were available for us to convert these low value renewable feedstocks like lignocellulose into these higher value products. And my lab, with no prior experience really in this area, decided to work with Uralia lipolytica. We took on the challenge of taking on a new organism, just basically because it had really interesting properties that make it a wise choice for trying to make oleochemicals. And in doing so and working with this organism that really hadn't had a ton of work done on it to that point, we realized that we needed to develop new tools, right? So this is where we bring the synthetic biology and basic things like bringing CRISPR-Cas9 into these systems and optimizing it so it works well, developing new promoters that are responsive, things like that are really important to us. To be able to help our lab and the rest of the field advance the engineering of these kinds of non-conventional organisms. And so we did this for a number of years, and started to get antsy and started to think about what else can we apply this kind of technology to. And so we started moving to even lower valued feedstocks, things that maybe are wastes and create problems for our society. So we've worked in agricultural wastes, animal wastes, human wastes, and more recently in waste plastics. And we've also at the same time started thinking about what broader class of molecules we might be able to efficiently make with biological systems. And so we've branched in away from, we still do oleochemical work, but we've expanded into natural products, enzymes, for example, and this sort of opened up a whole new line of thought for us. And so as we started to think about more of the feedstocks, the more complex, less well-defined, dirtier feedstocks, we started to realize that Uralia lipolytica can really only do so much. And so we started looking for other organisms to work with that had more favorable properties for working with more complex substrates. And so we've now started to work with a number of different hosts, which again has created the need for us to develop an entirely new suite of tools to work in these host organisms. The new tools we've also applied in other ways to problems in environmental sensing. And so again, thinking about how do we take engineer organisms out of the lab, put them in the environment to do hard work for us, like remediation, to detect threats, and doing so in a safe and biocontained way. So those are sort of some newer directions in my lab that you'll be hearing about more in the next few years. Our interest in things like enzymes, and again in the sort of the basic synthetic biology tool development led us into some projects in the mammalian cell space. Again, this is relatively new for my lab, and things that you'll see in the literature coming out in the next year or two. And the last thing I'll mention here is in thinking about how to deal with the waste feedstocks, we really have gotten deep into bioprospecting interesting environments like the gut of the yellow mealworm. And I'll say just a couple of really quick things about that, and then I'll stop rambling. So this is probably one of our more exciting of the many exciting things we do, projects. So we've been exploring the capabilities of the gut microbiome, as well as the mealworm itself, the capacity it has for degrading polyolefins, which are really difficult to degrade biologically. And so you can just see some of our data here. And the story is basically, we are still in the discovery phase. We're learning what microorganisms are in the gut and how they contribute in communities, as well as in isolation, to the degradation of things like polyethylene. We've identified some enzymes already from those microbes and also from the saliva of the mealworm that seem to have some degradative capacity. And so right now we have a lot of really exciting open questions, not a lot of answers, but we're really digging in now, and again looking for more people to join this part of our team. And so I'll end on this one slide. And I forgot to continue to make my analogies to making friends. And so we're sort of thinking about we've made more friends in the lab, trying to bring new organisms in. We're thinking actually now about how we build artificial communities to do complex things like break down plastic, like remediate the environment. And I wanted to point out that I think it's easy to get buried in all we have to do in research, or all we have to do in building companies that maybe some founders who have done this series before have done. But it's really important, at least it has been important in my career, and I think, Tae-Suk, you're probably in the same position. It's been really important to make friends and build communities within the synthetic biology, the technical community. And I've been fortunate enough to benefit greatly from my involvement in many organizations listed here. I've given a lot of time back to those organizations because I feel like they've been very important in me finding friends in a community that's really supportive. And I think that's one of the real strengths of synthetic biology, is that we have amazing people that all really support each other. And it's just really exciting to be a part of it. So I just have listed a few ways in which I've gotten involved, and I think maybe it can be a catalyst for some other people to think about ways that they can give back to our community, to our youth. And so I'll stop there and hand it over to I guess back to Tae-Suk, who will introduce Dave for the real talk today. So
that is amazing, Mark, as always. I absolutely agree with you. I mean, you've done great job to the community, sharing your time and service, everything. And I also love to see you answer one of my technical question about your plastic degrading thing, about saliva contribution and the bacterial contribution, sort of that. I'm glad. Also, you and I sort of share and then follow the similar path in terms of topics of research and everything. Also, the most important thing you mentioned is I was constantly asked by my university what is return on investment, but I answer always, our return should be our next generation student and postdoc. And so you mentioned that at the beginning. I really love what you mentioned. Thank you so much for your contribution to the society and your education effort so far. Thank you.
Yeah, thanks.
Yeah. All right. So now the main speaker of today with a longer introduction. Professor Dave Karig is pursuing tenure as an associate professor in the Department of Bioengineering at Clemson University. He's gonna submit the tenure package soon, but I believe he will get tenure soon. Prior to joining Clemson, he was a senior professional staff member at the Johns Hopkins University Applied Physics Laboratory. Before joining APL, Applied Physics Laboratory, in 2012, he was a postdoctoral researcher at Oak Ridge National Laboratory. He earned his PhD in electrical engineering at Princeton University, where he conducted his thesis work in the area of synthetic biology under the guidance of Ron Weiss, the last week's pioneer speaker, and he earned his bachelor in electrical engineering from Clemson University. That means he came back to his own university. Currently, his research focuses on synthetic biology and microbiome engineering. And I'm not sure he's going to talk about today his wonderful work about Turing pattern formation that published with the Ron Weiss several years ago. But among many, many work, I'm very excited to hear and listen to his talk today. And Dave, thanks so much for your time today, and please take it away.
Thank you. All right, and I really appreciate the opportunity to give this talk. So I'll mention the Turing pattern system, but I was mainly intending to focus on engineering cell communities, and that's a big focus of our lab right now. And in general, one of the things that always fascinated me stepping into synthetic biology, coming from electrical engineering, is just the tremendous diversity of natural microbes, the extreme environments they live in, the things that they do metabolically and sensing wise. And this talk is no exception. We're aiming to capitalize on the tremendous diversity of natural microbes and what they can do. But our goal here is going to be to build and shape microbial communities for applications, and that could be done with natural microbes or with through means of genetic modification. And we might imagine that if we want to develop a community of microbes for a particular application, we might start out by looking at the microbes that are there and sampling them and characterizing them and thinking of them as our candidate members. And if we can assemble a functional community, in general, the application spaces are about as diverse as the spaces you find microbes in, but we're going to focus on applications in the skin microbiome and in marine contexts. And taking a step back, most of us tend to think about synthetic biology in terms of perhaps starting with a set of well-characterized parts and devices. And typically these are genetic parts, perhaps promoters, ribosome binding sites, cell-cell communication devices, terminators, reporters, and so forth. And we start out with a vision, and through design, build, test cycles, and trying to apply engineering principles, we arrive at a prototype to sort of realize our vision. And this can manifest in different ways, but there's some commonalities across a lot of efforts. And to give an example, I've always been interested in emergent pattern formation. So you're starting out with a homogeneous lawn of cells, and they all have the same DNA, but a pattern emerges. So that's always fascinated me. And I particularly liked Alan Turing's theory of morphogenesis. And going all the way back to grad school, we tried to build a system inspired by that high level sort of network. But if you take a sort of high level network, perhaps a reaction diffusion network, but if you take that high level concept, there are many, many different ways of implementing that in a genetic network. And in this particular case, we used a fast computational method, linear stability analysis, to figure out which of those many, many implementations possibilities were enriched for the potential for forming patterns. And that allowed us to down select tremendously and then do more intensive spatial simulations. And then to build the system, as is commonly the case, we built it in a modular fashion. You establish your parts and devices, you interface them, testing along the way, and piece together the system in a modular fashion. But for today's purposes, I'd like to invite you to think about communities another way. So how do we move beyond E. coli? This is all in E. coli. How do we build multi-species communities that can work and be resilient in the application environments that we'd like to work in? So I'd like to kind of invite another consideration of another perspective of this. So here I'm referring to this as well-characterized species in the sense that we can maybe perhaps think of on a different level, different microbes of the community as playing specific roles. And we can imagine, a number of different functional roles. Like we might have, here they're called bullies, but perhaps they keep away unwanted members of the community. Reporters, perhaps they make a pigment that we can spot or see. Glue, we might have microbes that just help us stick to the right surface. We can imagine a lot of different functional roles, and we can think of specific genes even that might be associated with that. But of course, in a community, we may have roles that are simply thought of as keeping the community shaped correctly, keeping it together. But with that perspective, there are a number of needs. First of all, we likely want to characterize the space we're working in. Like, what are the resident or background microbes there? And then we have to get our buddies. We have to source our strains or candidate members that we want to potentially use. Once we have those, we need a way of assembling a community that's going to be stable. If you just throw microbes together, some of them are going to die out. They're not necessarily going to form a community. So we need assembly methods. And really, we want a more rigorous framework for design. So what does this design, build, test cycle look like? So the outline today is going to follow those topics. So first, I'm going to talk about characterization towards applications in the context of the human skin microbiome. Then I'm going to talk about community engineering efforts in the context of marine applications. So specifically in that space, we'll talk about sourcing strains. So perhaps if you want a specific function, how do you get there? Then, once we have our functions, how do we assemble a community of different species or members? And then I'll give a very high level idea of what a design method or framework might look like to actually help you do true design and co-optimize for all the things you need, your function and your resilience and so forth. So starting out with characterization, I had the opportunity to lead a MURI grant funded by Army Research Office, focusing on the human skin microbiome. And there are a number of reasons why we might be interested in it. First of all, certainly there are direct implications and impacts, wound healing, skin and soft tissue infections, but also the skin. It's a metabolic organ. It's very important for our, it's our interface to the world. But there are indirect impacts that are very interesting as well. So the microbes on our skin can shape, how we smell more or less. They shape our profile of volatile organic compounds. And that can impact the degree to which disease-carrying arthropods may be attracted or repelled. So our goals with this study were to, first of all, understand why do different people have different skin microbiomes? So what are the drivers of interpersonal variation? Moving to smaller scales, what is the fundamental scale of relevance or of change for the human skin microbiome? And then moving even further down, we'd really like to get down to the level of understanding what specific species do, and even molecules. So those are our high-level goals, and we started out through sampling and sequencing, as is common in microbiome studies. So we sampled a number of individuals in sort of 3-axis sampling. X and Y transects were done with swabs, and then we used successive tape strippings to get the Z dimension. And we use amplicon sequencing, so 16S rRNA gene sequencing for basically all the samples, and we did shotgun sequencing for a subset. But we wanted to go beyond this and really understand the differences and why we see what we ended up seeing. So we quantitatively measured a number of different skin characteristics and administered a very extensive questionnaire. And we don't have time to go into all the details of this. We're still trying to write this up, but I'll just mention a few highlights. One is that we found that lifestyle and demographic variables explain much of the interpersonal variation at a taxonomic level. So if you're looking at the mix of genera that appears on different people, really lifestyle is a big driver of that. But interestingly, lifestyle was found to explain, even be far more explanatory for functional diversity. So what does functional diversity mean? Well, basically, if we take those taxa, perhaps the genera, and map them into their traits or functions, then the picture can become much more clear. And we're going to come back to that point when we get to some of the engineering framework topics. But we want to step beyond this. This is a study, and a lot of the findings are correlation based. So how do we bridge those sampling and sequencing efforts to mechanistic studies and really applications? So a key part of that is to develop an isolates collection. And I'll credit Colin Tim for heading this effort. Also the smelliest effort I've been associated with. But Colin developed a number of different isolation conditions and basically sampled to the point where you're starting to see the genera, unique genera that we recovered saturate. And the collection is over 800 members. And it's been tremendously helpful for kind of opening up a number of directions. One is, studies of microbe environment and microbe host interactions. So here, Kristen Loomis, using those isolates, started out by identifying a number of interesting compounds found on the skin from mass spec literature. And she developed a high throughput method using tetrazolium dye, basically a redox assay that tells us which isolates can utilize which of these compounds. And that can be used for a number of interesting directions. One, you can tell which microbes might be competing for the same thing, so high niche overlap. You might be interested in knowing which microbes might grow more if you put a particular product on your skin. So some of these compounds are associated with hygiene and cosmetic products, or perhaps which might be modulating important host signals. So we also, of course, on that topic, really want to understand microbe host interactions. So you need a good model system, and there's no perfect model by definition, but we really like the human skin equivalent. So basically 3D tissue culture models. So we used epiderm models, which have keratinocytes and fibroblasts differentiated basically into an epidermal layer of the skin. And we can put whatever isolates or communities we want on these. And we did our analyses by transcriptomics and histology. And a high level finding is that microbes, particularly in a community context, have important impacts on host skin processes. And that has a number of ramifications in health and human performance. So to give a few examples, we found that Micrococcus can have a significant impact on epidermal thickness. So we're looking at this sort of purplish layer, the thickness of that. But also the mixed community had an effect on epidermal thickness. If we look at epidermal cell proliferation, several isolates impacted that, but the most pronounced effect was with the mixed community. And then we looked at two factors that are important for barrier function of the skin. So, if you're outside and there's, pollution, how much of it actually truly gets in you, for example. So filaggrin and loricrin are important for barrier function. And we interestingly didn't find individual isolates that significantly impacted those, but the full community had significant effects. So that's really kind of a neat indication that the effect of the microbiome is really more than the sum of its parts. And that in a way is actually kind of intuitive. So if you walk into a factory and sample some, but not all the worker classes, you're not going to get the correct factory product out of it. So I think it makes sense. But the take home here is that these microbes, the skin microbes, are really having very pronounced impacts potentially on the skin morphology and function. And the next steps there are a deeper dive into mechanisms. So we do have transcriptomics data, but it's for the host. And Lauren Elizabeth Franklin in my lab now is a grad student who's kind of chasing that direction through transcriptomics and host microbe transcriptomics, or metabolomics and host microbe transcriptomics. And then, after that point, when we have a better understanding of the precise molecular mechanisms, there are some interesting questions we can pose, like, can we optimize skin properties, for instance, barrier function? So, the take home here is it's characterization with a purpose. So those sampling results, they can tell us if we're going to develop an application. They might provide clues as to, is it going to work on 60% of people or 95% of people? And we can utilize the spatial results as well. So just arbitrarily, if you think of an example, like perhaps we want to make a probiotic sunscreen, you probably want to know if, the next day, if it becomes spotty and only on certain parts of your body, the microbes are persisting and, they're wiped out elsewhere. So those results alone are useful, But I think the mechanistic insights are going to be particularly powerful as we drive towards applications here. So now we're going to move into community engineering. And in marine spaces, in some ways, it can be a little bit easier to approximate that environment that doesn't require 3D tissue culture models. Although if we really want to properly replicate some aspects, it does get challenging, as we'll see. But our motivation here is initially we're focused on fouling. So fouling increases drag, lowers speed and fuel efficiency, and that can lead to pollution problems, slower shipping, more expensive shipping. So it's a long-standing problem, well over 100 years, and it's hard to address. And this work was made possible by an ONR Global X Challenge Award led by Nick Aldred at the University of Essex. And Nick is a barnacle expert. So our goal was to develop a microbial community to prevent macrofouling. And the approach was to embed specific functions in a rich and stable bacterial community. So I'm going to focus on our focus at Clemson, and in particular, I want to talk about a targeted function, and that's chitinase activity. So Nick had shown that chitin is a component of barnacle adhesive. But it's also a structural component of fungi and algae, and it really has a number of other applications as well. For instance, on agricultural pathogens, agricultural fungal pathogens. But here we're focused on the marine context, and there are two approaches we could take towards establishing chitinase activity. One is to screen natural microbes from the marine environment. So we can screen for natural microbes, and if we use those natural microbes directly, there might be simpler regulatory pathways to getting the application actually realized. Now, with genetic modification, of course, the benefit potentially is the flexibility. I mean, you can add functions, you can realize things that you're unlikely to find in nature. So we can bolt that chitinase activity onto sensors or other things. However, the caveat besides perhaps more regulatory barriers, optimization likely being required for really getting those engineered functions to persist. So they're both interesting, and they both have their relative strengths and trade-offs. So we pursued both. And I will say that the opportunity to work on this project with marine microbes and even doing sampling was very dear to me. So I grew up on a barrier island, Isle of Palms, off of South Carolina, and this was during COVID times that that this project started, 2020. And my father's a emeritus academician and a little bit of microbiology experiment. So I told him, hey, next time you come up to see your granddaughter, I need you to do one thing. So he brought up water samples and sent this charming, cherished email. And then we were also fortunate enough to have a wonderful postdoc, Dipti Chaligan, join us. And Dipti is now a research assistant professor working with us, who took this sample and identified a number of chitinase producing microbes. So that approach involved two things, isolating directly and then screening, but also doing enrichment, where we take some of the water sample and put it in chitin media, with the idea being that once all the other resources are exhausted in that media, you would hopefully enrich for the ones that can utilize chitin. And the screening method is based on chitin agar plates. So you see that sort of milky white area, that's chitin. And when the microbes break it down, you see those clearing zones. And of course, from that point, we can proceed to sequencing, identifying the microbes, identifying the genetic factors, and so forth. But before we get into that, I'll point out that it's kind of exciting to see chitinase activity continue on these plates for 3 weeks, and it might continue for longer. The plates just get a little bit dicey after 3 weeks. And I'll also mention you'll note some Florida samples. That's because Lauren Elizabeth in our lab said, "Can I go to Florida for spring break?" And the answer was yes, but I need you to do one thing. So we have South Carolina and Florida samples. They're taxonomically diverse, and the chitinase genes are diverse as well. So I should point out from this tree here of the identified chitinase genes from our sequencing and informatics efforts, that I should point out that there are different families of chitinase. And interestingly, all but one of the isolates that we recovered, all but one of them had multiple chitinase genes. And I'll also point out that chitinase activity, like the breakdown of chitin, is really more than just expressing a single gene. So all of the isolates that exhibited chitinase activity had genes from each of 4 classes. So binding chitinase, solubilizing it, depolymerization, and then further breakdown into monomers. And we wanted to move from this point. Now we can use the isolates directly. We know what they are. We actually profiled. We look for hits to the virulence factor database. So we can identify potential pathogens and rule those ones out. So we can move forward with natural microbes, or we could try and express these genes in other hosts that we might want to work with. So I'm going to talk about that latter direction now. And stepping into that, we chose Rhinamira SP to look into because it had a single chitinase gene. So we know that its activity mapped to that single gene. And first, characterizing it in characterizing the natural Rhinamira species and its chitinase activity showed that the chitinase activity was not really affected by adding other carbon sources like glucose, which can be a nice feature. Interestingly, when we added 0.1% colloidal chitin, a little bit of soluble chitin, that actually stimulated chitinase activity. So it seems to be sort of a sense and response mechanism, which makes a lot of sense. It's that can sort of help the bacteria navigate that cost benefit profile and avoid expressing chitinase in scenarios where it's not helpful. But moving towards heterologous expression, we chose a broad host vector. We integrated reporter genes just to make sure that we could express it in other hosts. So we use E. coli just because it's well characterized and well known, but we also optimized transformation methods for a number of bacteria from the Rosiobacter clade. And the Rosiobacter clade is highly prevalent and abundant in the ocean, and they're known for being good first colonizers. So they might be a good choice for a sort of design community. And we're focused here on one Rosiobacter denitrificans, but we were able to get reporter gene expression. So we moved on to integrating our chosen chitinase from Rhinamira, and we also developed some separate constructs just for comparison purposes using well-characterized chitinase genes from other organisms. And we started out by strongly expressing them, and basically nothing worked initially. So we're using a fairly strong promoter, the lambda PL constitutive promoter from lambda phage, and strong ribosome binding sites. So one logical thing to do was ribosome binding, basically tuning the expression levels. And by sort of optimizing the ribosome binding site strength using the SynBYSS lab tool, we were able to see chitinase activity. So this, lib1A2, lib2A1, we're starting to see chitinase activity. But what gave us the strongest activity was using the native promoter and 5' UTR from Rhinamira. And we hypothesized that that basically contains this sense and response mechanism. And surely enough, we saw our strongest activity in both LB and slightly less rich but defined M9 media, and was very encouraging. This is in E. coli, but we were able to establish at least 21 days of activity, just like in the natural system. So that was very exciting to see. And again, the plates just get a little bit questionable after 21 days. So it's really exciting to see that sustained activity through heterologous expression. Now, we were able to port this to Rosiobacter denitrificans through similar methods, the ribosome binding site optimization, and again, the native promoter and 5' UTR gave us the most pronounced clearing zones. And we also did some studies to look at when function was lost. And The main conclusion there, without going into great details, is it largely seemed to be due to plasmid loss and then genomic mutations that conveyed resistance to chloramphenicol selection. So we could likely further stabilize this through chromosomal integration. But the bottom line is we've gone from natural screen microbes all the way through heterologous expression that can last for a while. So that tells you a couple of different ways that you can get functions in your members. But how do you take those members and make a community? So our lab, there are different methods. Several groups have some very interesting and demonstrated methods. Our group likes this, what I'm calling shotgun assembly. So it's a top-down method where we take our sets of desired or candidate taxa, and we basically mix them all together, and we iterate through rounds of passaging. So basically, through successive rounds, we deplete away the unfit microbes that don't really, join and thrive in the community. And you can even select for resilience if your sort of depletion mechanism is a particular stress. But in doing this, in doing this several times, Al, we've kind of come across a number of interesting nuances to this, and they're actually really useful too. So to give one example, if you do this assembly process in rich media, you may very well identify what we're calling bullies, and that's probably shouldn't make up ecological terms, but kind of makes sense. So instead of getting a rich community after all this, we would often find just a single isolate, a single taxa. And these bullies are basically competitive first colonizers. They grow quickly, and they typically express strong antimicrobial competitiveness factors. So in the marine context, this would include a lot of the Pseudo-Ultramonas species. But one way to address this is if you're dealing with isolates, you just repeat the process and leave those bullies out. So that's one way of tackling it. But I want to give an example of what this process looks like. So we had the goal of trying to get microbes from a few different classes potentially together. So we considered bullies. We wanted to incorporate our chitinase producers. We tried several isolates that were just good biofilm formers that came from our collaborators on the ONRG effort at Copenhagen, Mette Bermela and Christina Amador. And we also tried to put in standard Rosiobacter isolates. And as we just saw, those can be transformed or genetically modified. So that was the basic idea. And the mixtures we put in just had the same CFU of each member. And in this case, we were looking at colonization of PVC. So we put little PVC coupons in a multi-well plate, and every day they'd be transferred to fresh media in a new well. So that's kind of how our passaging was done. Every day, the planktonic region was replaced. And I won't get into details for time limitations, but we developed a selective plating method. Basically on the right column, each of those numbers corresponds to a pair of inhibitors that help us to select for individual microbes. And we're also using pigments to help in cases where the selection chemicals aren't quite sufficient. And no selection method or quantitative community assembly method is perfect, but we're able to in some cases get down to strain level distinctions. So that's what we're using. Mainly, I want to show just some example results, focusing on this bullies issue. Or topic rather. So one is, we said that we often get these bullies when we use rich media, but what is the role of nutrients? Is it mainly a problem encountered in rich media? And can we actually build a community with these? So these Pseudo-Ultramonas have a number of interesting functions and features. Is it possible to build a rich community with them? So when we did this shotgun assembly and included these Pseudo-Ultramonas species in rich media, we just got 2-member communities out of it. So some of the Rosiobacters can hang out with the Pseudo-Ultramonas, but that's pretty low richness, 2 members. However, when we took marine broth and diluted it 100-fold to somewhat, roughly approximate, nutrient levels you might see roughly in the ocean, then we started to get more members. We got a 6-member community after a week. So next question is, can we build a community without bullies? And the answer is yes, and it's actually more diverse. We get 9 members basically, but if we build it without bullies, can it resist a challenge from the bullies? So this bottom right plot shows what happens if you take this community on the bottom left and you challenge it. So we're looking at days post challenge, and up to a week post challenge, it's not as though the community is falling apart. So that's just a way of getting very different types of communities. And there are a number of nuances that are kind of fun that we encounter as we go along as well. And I want to kind of finish up because just time limitations. I want to give a very high level idea of what a framework might look like for design. We don't want to just put things together and get stuck at a particular resilience or functional point. How do we co-optimize all the things we need to co-optimize? And this is a effort that we just started, funded by DARPA, where we're focused on engineering drag reducing biofilms. So earlier we talked about barnacles. Now we're actually just focused on the roughness of the bacteria, because that can actually have a significant drag penalty alone. And that's especially problematic for unmanned underwater vehicles, or UUVs. So these are basically like underwater drones, and they're commonly used in studying the ocean. You can go download sensor data from from these, and the duration at which they can operate is largely tied to how efficient they are. So all the current strategies really focus on anti-fouling, just getting rid of as much living matter as possible. But none of them really combine safety, effectiveness, and scalability. So our approach is to instead develop beneficial biofilms. So we call ourselves the eco-coding team. And our approach is to develop communities of natural marine microbes that form smooth and stable biofilms to reduce drag. So we are isolating microbes, or our so-called building blocks, from a biofilm from a UUV, and then we're going to use certain microbes to assemble a community that is smooth and stable. And to do this, the test bed, as I mentioned earlier, kind of alluded to that with the skin. We need good models, and the test bed will simulate the application environment and relevant perturbations. And we also need it to quantify function, in this case, morphology. And the data from the test bed can then parameterize models that guide our design and our design build test cycles. So that's a high level idea. And if this works out, then it's going to have a lot of extensions in a number of different areas. Even though we're focused on the specific case of drag on UUVs, we're looking first of all at a complex and emergent functional target. So it's not as though we're asking our community to do one thing like chitinase activity, or alter the concentration of a particular metabolite. Morphology really involves potentially all of the community members and the environment. So it's a complex target, and we're also co-optimizing for resilience, invasion resistance, and overall duration of this. So if that works out, it's going to give us a framework that's pretty broadly applicable. But back to the specifics of kind of how we're looking at this, we're making parallel plate flow chambers to look at, or kind of recreate, the perturbations these biofilms might experience through changes of speed, for instance, of a UUV, or diving, which could affect temperature. You decrease as you dive. And we're also quantifying morphology by optical coherence tomography. You've probably had that done to your eyes at the eye doctor. It's just a really fast way to look at morphology. And we're also integrating superspectral imaging. So the model is led by Ling-Chong Yu at Duke. So yeah, I'm not going to go into much detail. I'm just going to mention that I think it can be very helpful to kind of merge parametric models and machine learning. So the parametric models have advantages like helping you extrapolate and interpret, but machine learning can be very helpful for tackling just the breadth of this problem, the number of possible interactions between your community members and the environment. So I'll just mention that at a high level. And then earlier I mentioned trait-based analysis and functional analysis and how that helped us to kind of understand the drivers of changes in the human skin microbiome. But tying this to engineering directions, we're going to characterize specific sets of microbes or isolates and make predictions. We really want to broaden our model predictions and project beyond the characterized taxa and kind of elucidate broader design principles. So a first step is to build a database of traits and genomic features. So we're using isolates. We can we can sequence them. We can actually do genomic characterization. And then using that database, that's going to allow us to map our isolates and the sets of them that appear in communities to trait space. Then we can predict performance characteristics as a function of trait composition. And that can give us that broader understanding. And then perhaps we can think about inverse mapping, like mapping that back and even making predictions for taxa that we haven't directly used or other locations. So, building, we already kind of talked about isolations and we gave the extensive example of chitinase activity, but Marc Gué at University at Pompeu Fabra University in Barcelona is leading the charge on doing the screening and selection for a number of different functions. And then we talked about a shotgun assembly, but Meta Burmala and Christina Amador have for years successfully done a complementary bottom-up procedure. So that's how we're building. And then I'll just mention at a high level, over time, we can achieve our goals by starting out with perhaps characterizing a stable community under static conditions and understanding the drivers of morphological changes, smoothness, roughness. Then we can apply perturbations with our test bed and start to optimize for resilience. And from that point, we can just sort of characterize a broader and broader space of taxa and microbes. And that's going to give us more design options, really, as we try to co-optimize all these things. And one of those things we can co-optimize at that point would presumably be invasion resistance. So that's kind of high-level description of a framework. And to kind of summarize and talk about next steps with the skin microbiome, we can, I think, see how the characterization can help with applications and seed us in those efforts with the isolates collection and an understanding of what may or may not work on different people. And the next step is, as I mentioned, to kind of characterize the molecular underpinnings of microbe-host interactions. Chitinase, the efforts show how we can go from samples to natural microbes that we can use, but carrying that all the way through to heterologous expression that's sustained for a decent amount of time. With shotgun assembly, it can be used a number of different ways. We just kind of touched on it, but we can target membership, we can target levels of richness, and we can select for specific resilience directions. And then the last step for drag reduction, again, just gives an idea, kind of seeds an idea of how we might develop a true engineering framework. So with all this in mind, again, thank you for the opportunity to give this talk. And I have a lot of people to thank. I did my best to get the right names in the talk. I'm sure I missed a few, and I have really a lot of people to thank in my group on our skin microbiome URI effort, ONR Global X Challenge led by Nick Aldred, and of course our DARPA eco-coding team. So thank you, and I'd welcome any questions you have.
Absolutely wonderful talk. I also glad seeing Marc Gué and Ying-Chung Yu. They are amazing scientists, and then they're part of your team. So I want to give the opportunity to Marc saying something because he needs to leave sooner than later. So do you have any comment or question?
I was just gonna say thanks for everything. Thanks for a great talk, Dave. I really enjoyed it. I'm really excited to see the continuing building of the direction of your lab. You're doing some really amazing stuff that I'm really jealous that we're no longer working directly together. But really excited about these directions, and wish you all the best.
Thank you. Really appreciate it.
And sorry, I gotta duck out now. Thanks.
Sure. Thank you so much. I'll ask some question while waiting for a question from the audience. So the one thing kind of stuck me the most is basically you basically try to engineer microbiota. That's one of my, huge interest. And in terms of, building the community, there are two different ways to do so. One is basically you start with the, basic, component. I believe that's your approach, kind of, and then assemble them together to make a synthetic community. And the other approach would be you start with the native, whatever, community, and then, sort of kind of use some microbiome engineering tools. I mean, including my own tool we recently published. Basically you can now kill one component, one guy from the, you can kill the bully, for example, using the CRISPR whatever method, and then you can kind of see whether that bully's death kind of contributing better or something. But there are two approaches. But your approach, I believe, the synthetic, building up, or from the bottom up type of approach. And then, is there any kind of special reason to do so, or I mean, you kind of also considering both of them?
Yeah, I mean, I think really considering the combination is really the best bet. And just to make sure I understand the question, are you referring to like how we focused on member by member and function by function? Yeah, I mean, I think there are definitely advantages to using microbes from the environment. I mean, certainly we can convey certain stress protections through genetic modification. I think there's quite often, a lot of things that that we often, that are easy to overlook, I suppose. Like, perhaps we think that, this microbe we want to work with, if we want to put it in the ocean, we just have to somehow get its salt tolerance up. But, quite often there are other factors there that are easy to overlook. But I think really the kind of combination of top down and bottom up is almost necessary.
Sure.
Yeah,
So,
because oh, sorry, good.
Oh, go ahead.
Oh yeah, because I was gonna say, like, if you're just building through the bottom up method, then you may eventually encounter more problems with, say, invasion. But if you just do top down, then you have to kind of go back and understand the how the community actually assembles. So really kind of the combination.
I see. So if you choose the bottom up, I guess you also explained that approach as well. Are you thinking about some genetic modification of those microbes?
Right. I mean, that's I suppose it depends on the precise application. Are we going to, if is it something that we're going to use out in the open environment, or is it something that's we're going to put in a fermenter? So I think the application context really determines what's optimal, I would say. I'm interested in both, but I have to say that really it's a really intriguing idea to me to ask, like, just how far can we go with natural microbes? I mean, because they're fascinating. So how can we as engineers combine those parts, so to speak, and achieve defined behaviors? So it's a that's a really fun challenge that's really sticking out in my mind. But I'm a synthetic biologist, and I'll always be interested in the other side as well, genetic modification. So really both. I know that's.
Yeah, I mean that I asked the question constantly to me because the reason I asked the question is, if you modify an organism, you need to deal with the biocontainment issue. So that's why I'm asking that question. So I guess you aware of that issue, and then that's why you kind of wanted to kind of use the mixed kind of approach, both native bacteria, and only just use the combination and assembly and the community building. And then the other one is also modification genetically those bacteria, but still, you probably to make sure the biocontainment ensured, I guess. if you're thinking about deploying those bacteria into the environment. So that's why I'm asking that question. Let me see whether we have question from the audience. I don't see, and also I see my clock 11:04, and I guess we just close and then chat more. Informal kind of discussion after the talk. Let's see. I didn't see any question. Okay, so let's close. So thank you all for joining and staying today. We'll meet again next week on August 31st. Oh my God, the summer is over. Thursday, the same time, the same Zoom link. We'll have Professor Mark Isalan at Imperial College London and Dr. Dave Garcia at DoD DevCom CBC. Interestingly, another Mark Dave duo will be the seminar speaker next week. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I'm stopped recording. Just give me 1 second.